This is chapter 25, which is really about the origin and diversification of eukaryotic organisms. In particular, this chapter goes over eukaryotic supergroups and the organisms that we loosely classify as protists. We also talk about endosymbiosis as part of the origin of eukaryotic organisms. So eukaryotic organisms in general have organelles and are structurally more complex than prokaryotic cells. They have a nucleus, they can have mitochondria, they can have chloroplasts. They have well-developed cytoskeletons that enables eukaryotic cells to have more complex shapes and asymmetrical forms than prokaryotic cells. Eukaryotic organisms can be unicellular or multicellular. There's a lot of diversity in eukaryotic organisms. They have all kinds of different nutrition, broadly speaking, a plant, an animal, fungus. Those are all eukaryotic organisms, and they have different kinds of nutrition. Size varies as well, right? There's single-celled eukaryotic organisms, and there's blue whales, which are huge eukaryotic organisms. And reproduction varies. If you remember from Bio 1, right, there was those different life cycles and sexually reproducing organisms like haploid, diploid, alternation generations, and some eukaryotic organisms do asexual reproduction. So it really just depends on the kind of organism that we're dealing with. With eukaryotic evolution, there's chemical evidence for the presence of eukaryotic organisms 2.7 billion years ago. That is after prokaryotic organisms, right? Prokaryotic organisms, there's evidence of them up to 3.5 billion years ago. The earliest fossils of eukaryotic cells are 1.8 billion years old, and the initial diversification of eukaryotic organisms occurred 1.8 to 1.3 billion years ago. With eukaryotic evolution, there was an appearance of novel features like complex multicellularity, sexual life cycles, and eukaryotic photosynthesis that arose 1.3 billion to 635 million years ago. Don't memorize those numbers, but I do want you to know, broadly speaking, this is later than prokaryotic organisms. Molecular analysis really indicates that eukaryotic organisms are kind of combination organisms, meaning they have genes and cell characteristics that are derived from both archaea and bacteria. This is likely a consequence of endosymbiosis, which is a symbiotic relationship in which one organism lives inside the body or cells of another organism. So we talked a little bit about this in the previous chapter when we talked about those alpha proteobacteria and cyanobacteria. So the endosymbiont theory proposes that mitochondria and plastids were formerly small prokaryotic organisms that began living within those larger eukaryotic cells, or what we now know today as eukaryotic cells. They could have been undigested prey or internal parasites, but over time, the organisms combined and became a single organism, right? So our cells have mitochondria. Those are now our mitochondria, and that's part of us. We're a single organism there. So some key eukaryotic characteristics and their inferred origins. So the DNA replication enzymes are very similar to archaea. Transcription enzymes, what's transcription again? You got transcription and translation. Both of those are part of protein synthesis. That's right. So transcription is from DNA to RNA. Those enzymes are archaeal. Translation, that's RNA to protein, mostly from archaea as well. Cell division, very similar to archaea. The endoplasmic reticulum, remember the ER? That's similar to both archaea and bacteria. Mitochondria, that's bacterial. Metabolic genes, well, where's most of our metabolism happening, right? We're making our ATP mostly in that mitochondrion, so that's mostly bacterial as well. As far as kind of broad evolution of eukaryotic organisms, the idea is that there was an enfolding of the plasma membrane, and that surrounded the DNA and chromosomes. So that's an early nucleus, right? And then more enfoldings of the cell membrane became those membrane membranous organelles like the endoplasmic reticulum. Then this big honkin' cell took in another free living cell, so it engulfed an aerobic bacterium and kept it. And that aerobic bacterium became the mitochondria as we know it today. And then that endosymbiosis happened again for some organisms, and they engulfed a photosynthetic bacterium and they kept it, and that is similar to modern day chloroplast. So there's lots of supporting evidence for the endosymbiotic origins of mitochondria and plastids. The inner membranes on mitochondria and plastids are similar to the plasma membrane of prokaryotic organisms. The division of mitochondria and plastids is similar to the division in prokaryotic organisms. And again, that DNA structure inside of mitochondria and plastids, that's right, mitochondria and plastids have their own DNA, and it's similar to prokaryotic organisms. Also, mitochondria and plastids can transcribe and translate their own DNA to make their own proteins. They also have their own ribosomes that are more similar to prokaryotic ribosomes than eukaryotic ribosomes. DNA sequence analysis indicates that mitochondria arose from alpha proteobacteria, and plastids likely descended from a single common ancestor, a cyanobacterium, that was engulfed by a eukaryotic cell. So here's a picture of endosymbiosis again, where, a cell took in another cell. So with primary endosymbiosis, that's when a heterotrophic host cell captures a smaller cell by phagocytosis but doesn't digest them. And then with secondary endosymbiosis, that's when a eukaryotic host cell ingests and retains another eukaryotic cell that already had one or more primary plastid. So plastids of some green algae contain this tiny vestigial nucleus that's called a nucleomorph. So we have evidence for that as well. So with multicellularity happens in a variety of forms. So there's kind of the second wave of diversification of eukaryotic organisms that occurred when multicellularity evolved. The first multicellular forms were probably colonies, which were collections of connected cells with little or no differentiation. So simple filaments, so long chains of cells, or balls or sheets of cells. These cells were held together by shared cell walls or connecting proteins. Later, the cells differentiated and got specialized kind of structures and tasks that they were doing. And really, this transition to multicellularity may have resulted from changes in how genes were expressed or used rather than the creation of lots of novel or new genes. So this picture just kind of shows the evolution of multicellularity in photosynthetic organisms, here with kind of a group of algae, like there's a single-celled organism here, and then gonium has multiple cells, and then there's kind of a ball of cells, and then Volvox is sort of an interesting organism, and it's got multiple cells and also an extracellular matrix, so material that those cells secrete from themselves to build up. In the other side of the image, we've got sponges, which are really kind of primitive animals there, and many of the cells in the sponge are very similar to a chanoflagellate protist there, so you can kind of see this like loose association of cells, then you get a more complex association of cells in other animals. So that brings us to the protist. So I put little scare quotes around protists because protists as a grouping is a very loose group, and our evolutionary understanding of relationships between protists and other organisms is still kind of in flux. Classically, protists include algae, which were plant-like organisms, protozoans, which were animal-like, and fungus-like protists like slime molds and water molds. However, protists are no longer recognized as a eukaryotic kingdom because they're not a monophyletic grouping, meaning they are not more closely related to each other than to other organisms. So to kind of remind you again, this purple box here is a monophyletic grouping, right? You've got the ancestor and all of the descendants in one grouping. That's a correct grouping based on evolutionary history. A paraphyletic grouping is missing some of the descendants, and a polyphyletic grouping is missing the ancestor. So we're saying protists are not more closely related to each other than they are to other organisms. So there's really no such thing as the kingdom Protista anymore, because we just found that not to be true. So which two eukaryotic organelles arose through endosymbiosis? That is the mitochondria and chloroplast. And then for number 2, which of those pictures has correctly highlighted monophyletic clades? I'll give you a moment to look at that, or you can pause it if you want. So our first one is a monophyletic grouping, right? We've got the ancestor and all of the descendants there, so that one's okay. Second one, not okay. This third one actually is okay, because we've got the ancestor and the descendants there. This fourth one, we're missing the last one, so fourth one, not a monophyletic clade either. So again, we can kind of classify what we call protists by their ecological role. Algae are photoautotrophic, or those plant-like protists. Protozoans are mostly heterotrophic, so they're animal-like. Then slime molds and water molds are heterotrophic, but they're more fungus-like. They tend to be, they can be decomposers. Some of them also ingest their food. And again, these terms are widely used. Like, people describe photosynthetic single-celled organisms and photosynthetic multi-celled organisms that aren't plants as algae, but that term algae is more about ecological role and doesn't talk about like evolutionary meaning for the most part. You can also classify protists by motility, or how they get around. Flagellates use flagella. Ciliates use cilia. Amoebas use pseudopods, which are these kind of like false feet, like little extensions here, are pseudopods. But it's not like these pseudopods all stay right here like weird little feet or legs. They can actually do cytoplasmic streaming and shoot pseudopods out other parts as well. Other protists get around by like gliding on protein or carbohydrate slime as well. So these are not the only way protists move, but these are common ways protists can move. So let's classify this pictured heterotrophic protist by motility and ecological role. So if we look at this one, we said it's heterotrophic, so it's not going to be algae, and then it has these little teeny tiny hairs up there, right? So this is a ciliate protozoan. The next one, it's a autotrophic protist, and we're going to classify it by motility and ecological role. So if it's autotrophic, it's algae, and it's got this tail here that's long, so that's a flagellum. So that is a flagellate algae. So that brings us to eukaryotic supergroups. So the 4 eukaryotic supergroups are proposed based on both morphological and molecular data, and the current hypothesis divides all eukaryotic organisms, so plants, animals, fungi, and protists, into 4 eukaryotic supergroups. These supergroups are Excavata, SAR, Archaeplastida, and Uniconta. There's still work to be done, though, with this protist classification and eukaryotic organism classification, because currently all 4 supergroups are dividing simultaneously on this phylogenetic tree. So that can't be correct, but we're going to revise it as we get more data available. So if we look at our different supergroups, so I want you to know the different supergroups, and I'll kind of tell you what I want you to know for the test, and then I'll tell you some things that are, just sort of examples to show you some diversity of life. So I want you to know the supergroup excavata is named for an excavated feeding groove that's in many of the cell representatives. Not all members have this feeding groove, but the ones that do, they can take food particles in by phagocytosis, which is a type of endocytosis. Here's a couple examples that I don't want you to memorize, but in lab you're going to see euglenozoans, which can include predatory heterotrophs, photosynthetic autotrophs, and even parasites. If you've heard of Giardia before, the organism that causes that is in the supergroup excavata. And then also, if you've heard of Trich, the vaginal infection, the organism that causes that is also in excavata. But again, don't memorize those. What I want you to know for excavata is that it has an excavated feeding groove. So then we have the supergroup SAR or SAR. This is a kind of diverse monophyletic supergroup, and it's named for the first letter of its 3 clades that make it up. So I want you to know supergroup SAR is made up of straminopiles, alveolates, and rhizarians, right? That's what the S, the A, and the R stand for. Straminopiles are named for having kind of this hairy or straw-like flagellum. Alveolates are named for having sac-like membranous vesicles or alveoli. This is kind of cheesy. Oh, literally cheesy. In one of my classes, somebody said they remembered alveoli because it sounds like ravioli, which is like a pasta sack. So alveolates have alveoli, which are sac-like structures. And then rhizarians, the root word rhiza means root. Rhizarians are named for these phyllopseudopods, which are thin, root-like. Well, somebody decided they were root-like. They're not like root-like a plant's roots in reality, but they're thin pseudopods and not thick pseudopods like we'll talk about later. So you can see in this image on the slide at the bottom, there's a couple examples of phyllopseudopods in the rhizarians there. So again, the thing I want you to know about straminopiles is that they are part of SAR and they have those straw-like flagellum. Don't memorize these examples here, but a couple examples of straminopiles are diatoms. Diatoms are kind of neat. They have these glassy cell walls. That picture of the diatoms there is just showing their kind of glassy cell walls, and this is actually artwork that somebody did where they arranged these diatoms. This is another straminopile, brown algae. Brown algae are the largest and most complex algae. They're multicellular, most are marine. Things that you might call seaweed could be brown algae. In the alveolates, again, the thing I want you to remember about alveolates is that they're part of SAR, they're the A. They have those alveoli, those sac-like structures. So don't memorize these 2 examples, but there's dinoflagellates, which have 2 flagella, right? 2 flagella. They're responsible for harmful algal blooms or red tides. They also are these cool bioluminescent algae. This is like, I don't know if you can still buy it, but there was a while where you could buy the dinoflagellates in this little dinosaur holder there. And I used to grow them in my office and bring them out to show them because it's kind of cool. But the bioluminescent part is pretty short term, and so like by the time I would get the container of bioluminescent algae down to the classroom and then shake it, it would be super brief. So I ended up stopping growing them. And then ciliates are another example of alveolates. Most ciliates are predators of bacteria and other small protists. So then we've got the rhizarians, which are, again, the R in SAR, and they are named for their phyllos or thin pseudopods. So many of them have those phyllopseudopods. Some of them have these kind of porous shells that are pictured here that are called tests. But don't memorize that. Rhizarians, phyllopseudopods, that's what you need to know. Part of the SAR. That brings us to the supergroup Archaeplastida. So Archaeplastida, the thing to know about them is that they acquired a cyanobacterial endosymbiont as a plastid. So I'm not going to trick you and have you separate, brown algae from red algae and green algae. But Archaeplastida, know that they have plastids. So red algae is an example of something that falls into the category Archaeplastida. They are red because they have an accessory pigment that tends to make them more reddish in color. Green algae are also archaeplastids. They have chloroplasts that are more similar in structure and pigment composition to those of land plants. And then I do want you to know that land plants fall into Archaeplastida because we're going to talk about land plants in another chapter as well. So Archaeplastida has plastids, includes land plants. That brings us to the supergroup Uniconta. Unfortunately, they don't have like a catchy little phrase for them, but Uniconta includes two clades, the amoebozoans and the opisthokonts. I want you to know those two clades. So opisthokonts includes animals, fungus, and related protists, and support for the close relationship between these clades is kind of mixed, and the research remains a little bit controversial. So in different books, they might separate these out a little bit differently, but we're going with our book here on this one. So in that supergroup Uniconta, there's the clade Amoebozoa. So amoebozoans have the kind of lobe or tube shaped pseudopods rather than the thin phyllopseudopods that the rhizarians have. So most are heterotrophic. They can feed on bacteria and other protists. Some also feed on like detritus, which is sort of kind of rotting plant material. There's also cellular slime molds, which are really kind of an interesting organism. They were once thought to be fungi because, like fungi, they produce kind of fruiting bodies and produce spores. However, genetically, they're not similar to fungus. So DNA sequence analysis indicates that the shared trait of fruiting bodies and spore-producing structures is really something that came about through convergent evolution rather than direct ancestry. And really the life cycle of slime molds kind of raises the questions regarding the nature of an individual organism. So I'll show you that more on the next slide here. So this slime mold has kind of a cool life cycle, right? They have a solitary single-celled amoeba phase here that can feed, and then those amoebas can aggregate together and form fruiting bodies which produce spores, which then grow up to become more solitary amoebas. But those solitary amoeba can also get together and do sexual reproduction to produce more amoebas. So really kind of a neat organism there, and you'll see some of that in lab too. So that brings us to the clade Opisthokonta in the supergroup Uniconta. Opisthokonts are a diverse group. They include the chanoflagellate protists, which are similar to the cells that are found in sponges, animals, which we'll talk more about later, nuclearia, which are protists, and fungi, which we'll talk more about that later as well. So fungi and animals fall into the clade Opisthokonta, which falls into the supergroup Uniconta. So let's review then, match the specific example to the eukaryotic supergroup. If you need to pause it, feel free to do so. For me, I would probably start with what I would think is the easiest one for me. And I know like plants have plastids, right? So Archaeplastida is this example here. We just went over the group that has the amoebozoans and the opisthokonts, and this is like an amoeba with, they're not called normal pseudopods, they're just called pseudopods, but there's like thick pseudopods, right? So what group includes amoebozoans and opisthokonts? That's Uniconta, so that is C. So, another one. I'm going to say, another easy one. Of course, it's easy for me, right? But another one that might be kind of easy has the Straminopiles, Avulots, and Rhizarians in it. So an S, an A, and an R. So that's going to be B SAR for that one. And so then process of elimination, we've done A, we've done B, we've done C. So that means our first one with the euglena is excavata, which was D. So, broadly, why do we care about protists? I mean, they're important. So in aquatic communities, photosynthetic protists, and prokaryotic organisms are the main producers. So I think when people think about producers, they think plants, but algae are really important producers as well. Some protists are in symbiotic relationships with hosts, and they benefit their hosts. So in coral, there's dinoflagellates that are part of coral. Maybe you don't care about termites too much, right? But there's wood-digesting protists that digest cellulose in the gut of termites. Some protists are in symbiotic relationships that are parasitic. If you've heard of sudden oak death, that's caused by a protist. If you've heard of potato blight, that's caused by a protist. If you've heard of malaria, that's caused by a protist as well. So protists have all different kinds of roles in ecosystems, and some are beneficial to organisms they're in close association with, some are harmful. It just sort of depends on the protist. It's a pretty broad group. So to kind of tie together chapter 25, we talked about the origin of eukaryotic organisms, right? So we had a large cell that had an infolding of the cell membrane that surrounded the DNA. That was the nucleus. More of that infolding of membrane became the endoplasmic reticulum. That kind of big chunky cell took in other free living cells. That's endosymbiosis. So it did that with alpha proteobacteria. That became mitochondria, and another line of cells did it again with cyanobacteria, and those became chloroplast. And then some big free-living eukaryotic cells took in other free-living eukaryotic cells and kept them. So that's secondary endosymbiosis. And really, all of that endosymbiosis and horizontal gene transfer really complicates our understanding of kind of evolution and ancestry and those sorts of things. So with multicellularity, it began as sort of simple colonies with filaments or sheets of cells or balls of cells. And then we got kind of more complex from there. And then as far as protists go, like that term really doesn't have any evolutionary meaning, but something is a protist when it is eukaryotic and not a plant, not an animal, and not a fungus. So we can describe protists by their ecological role. Algae are photosynthetic. Protozoans are animal-like and heterotrophic, and then mold-like protists are also heterotrophic, but more similar to fungus than animals. Then with our eukaryotic supergroups, we've got excavata, which is named for having an excavated feeding groove. We've got SAR, which is named for the stramonophiles, aviolates, and rhizarians, the clades that make up that group. Stramonophiles are named for having straw-like or hairy flagellum. Aviolates are named for having alveoli, which are those kind of membranous sacs. And rhizarians are named for the root-like, thin phyllos pseudopods. We've got Archaeplastida, which are named for having plastids. That includes red algae, green algae, and plants. And then we've got Uniconta, which doesn't have a catchy, cute little phrase, but Uniconta is broken into the amoebozoans and opisthecons. Those are two clades that make that up. Amoebozoans are organisms that have pseudopods, but not the thin pseudopods like rhizarians. And then opisthecons includes fungi and animals. So that's it. We made it through chapter 25.